right, we are in hour 5. I will bring up these slides here. And it only gets more exciting as we go on into the evening. So here we have a panel with different presenters from many different labs, all being interested in single cell studies and also connected via a project where we are trying to understand cell distances to vasculature across different tissues. And this is in support of using the human vasculature as a common coordinate framework to get us a latitude-longitude system for the healthy human male and female bodies. And it's super exciting to get so many of you on one panel. And I will just go through these slides, or some of you might like to present your slides yourself. But let's see if we have everybody. Chenchen, are you here? Maybe I just stop sharing for a second to just see who is now all in here.
Yeah, I'm here. Maybe can you give me 2 minutes? I'm still trying to set up my camera.
Yeah.
Or someone maybe can go first. I don't know if that's easier.
Absolutely. And
That will
since
be easier for me. Yeah.
you're here, Kevin is maybe joining a little later. Archibald is here. Good to see you, Archie. Alex is here. And Ravi is also here. So we have a lot of different tissues and a lot of different expertise from major labs in the US. So in earlier presentations, we had people from all around the globe. This is a very US-focused event funded by different NIH projects. And so all of us are coming together to try to see if we can potentially at some point later use the vasculature as a common coordinate framework. So I can share these slides. I'm happy to do this, Chenchen. Or we go to a different presenter. Kevin is here now also. Hi, Kevin. Good to see you here.
Yeah,
So,
it will be easier if someone can jump for my slot. So I'm trying to get another laptop to set it up.
okay.
Yeah.
All right. So let me go on to the second presentation, which is Sam. Sam, would you like to share yourself, or should I share your slides? Happy to do either.
Yeah, you can go ahead and share. That's perfect.
All right.
Thank you so much.
And some people had a slight delay. I guess I'm at the lake here, so that is not the best internet. All right, Sam comes from the University of Florida, works very closely also in the HubMap project. And it's just a pleasure to work with you and your team, Sam.
Yeah, thank you so much. Well, as Dr. Börner said, my name is Sam Ewing. I am a scientist at the University of Florida. And today I'll be presenting on our work on the human pancreas as part of a larger effort within HubMap to do multi-omic spatial mapping for the human pancreas. This has been in collaboration with Pacific Northwest National Lab and Texas Advanced Computing Center. And so the work I'll be presenting on today has kind of been born out of that collaboration. So next slide. Yeah, so I'll be specifically talking about using the tools of the vasculature common coordinate framework on the human pancreas. And yeah, very excited to share our work with you today. Next slide. So just to give a brief introduction to the human pancreas is an organ that sits right next to the duodenum, and it's composed of two main portions, the exocrine and the endocrine portions. The exocrine portion accounts for about 95% of pancreas volume, and the functional tissue unit there is called an acini, which is composed of acinar cells, which secrete digestive enzymes into the digestive tract. And then the endocrine portion is a much smaller portion of the pancreas, and it consists of functional tissue units called islets of Langerhans. And these cells secrete hormones into the blood vessels and perform some very important functions in the body. Next slide. So to look a little bit closer at one of those islets, you can see here in the image on the left, the three main cell types in islets are alpha, beta, and delta cells, which secrete the hormones glucagon, insulin, and somatostatin respectively. And insulin, as you may be familiar with, is important in the regulation of glucose metabolism. And so our lab specifically studies type 1 diabetes, which is an autoimmune disease of the pancreas, where the immune system attacks these pancreatic islets, kills primarily the beta cells. And since beta cells are responsible for insulin release and glucose metabolism, this leads to glucose dysregulation in the body. Next slide. So our lab has been working to understand the pathology of type 1 diabetes in more detail in collaboration with many other excellent researchers. And back in 2019, there was a paper that came out looking at microvascular alterations in type 1 diabetes. So in this image on the left, you can see a couple control islets that look healthy with all the insulin containing beta cells. And then in the images at the bottom, we see islets in type 1 diabetic patients where those beta cells have been lost. And basically what the paper found was that there was a decrease in the vessel diameter within those islets of type 1 diabetic patients, but there was an increase in the density of blood vessels. And so this paper was one of the things that kind of motivated us to use the tools of the Vasculature Common Coordinate Framework to be able to look at some of our data and see if we can elucidate anything more about changes in vasculature within type 1 diabetes. Next slide. So to do that, we work with pancreases that are acquired from organ donors. So far, we've analyzed about 8 pancreases from HubMap that are control healthy patients. And then we've analyzed about 12 pancreases from another study. And within that group, some are type 1 diabetes patients, and then some are autoantibody positive patients, which means they have autoantibodies that may ultimately lead to them developing type 1 diabetes later on. So basically, we performed multiplex immunofluorescence staining as part of spatial profiling using nanostring genomics technology. And the markers that we looked at in this first study were insulin to mark the beta cells, pan-CK to mark the duct cells, and then CD31 and 34 to mark the vasculature. Next slide. So to do the analysis, I've been using QPath software. QPath is an excellent open source software for bioimage analysis. And within QPath, I basically developed an analytical pipeline to first do tissue detection and then do single cell detection. And using thresholding, we're able to characterize each single cell within the pancreas as positive or negative for each of the protein markers that we looked at. And then that data gets exported from QPath as a CSV file. And then with some proper formatting, we're able to put that into the Cell Distance Explorer, which is an excellent tool that Hive has developed and is continuing to develop. And that allows us to really look at the data spatially in 2D and look at those distances from the different cell types we have to the nearest endothelial cell or vasculature. Next slide. So these are a couple of the images that we got from the Cell Distance Explorer. This is showing a rendering of the pancreatic islets. And you can see in the top 2 images from the control patients and the autoantibody positive patients, we see pretty normal, healthy islets showing a bunch of beta cells. And then those lines are showing the connections to the nearest endothelial cell. And then in the image at the bottom, we can see a patient with type 1 diabetes where there's beta cell loss in the islet, much fewer beta cells. And we're hoping to see whether we can detect any differences in the spatial biology and maybe like the distance to the nearest vasculature in those patients with type 1 diabetes. Next slide. So these are a few of the graphs that we've got so far. The ones on the left are from the HubMap control pancreases, and we've been working with Yash from Indiana University, and he's been excellent at helping us analyze this data. And there will be a publication coming out soon, so I won't go too into that. But the graphs on the left kind of show the differences across regions of the pancreas, just within control healthy patients. And then in the graph on the right, this was some of the more recent data that I've been looking at, where we can see the 3 different groups of control on the left, the autoantibody positive donors in the middle, and then type 1 diabetes on the right. And you can see that it looks like there's a trend towards shorter distances in the type 1 diabetes group. And this would be in agreement with the paper that I mentioned earlier that saw kind of an increase in the density of vasculature within the islets. That would probably shorten that distance a little bit to any of the beta cells that are still there. And so with what we've done so far, we haven't found any statistical differences, but it certainly seems like there may be a trend within the data that's worth investigating further. And so we're kind of just getting into this and very excited to continue analyzing new data using these tools from the Vasculature Common Coordinate Framework, and hoping to be able to that will shed light on type 1 diabetes pathology and perhaps changes in the vasculature that may be present with the disease. Next slide. So with that, I would like to thank my PI, Dr. Martha Campbell-Thompson. I would also like to thank Dr. Fu and Dr. Cates from our lab, who have been very helpful with this. I would like to thank Dr. Börner from this call for all of her help and guidance with using these tools and implementing these tools, as well as Yash from Indiana University and all of our other collaborators. So thank you so much.
Great to see this evolve over time, see the data come into existence, see first analysis, visualizations happening. Yesterday we had the 8th HRA release, so we now have a new cell distance explorer that you might like to try out. Many of you had feature requests and we implemented some of them, not all of them, there were too many. So please check it out. All right, in the interest of time, try to keep it to 5 minutes so that we do have time to have a discussion. So Chenchen, are you ready?
Yeah, anytime.
Please.
Can you show the slide?
You should be able to share your screen, yeah.
Oh, do you want
Or
me to share?
I can also share, right?
That will be easier.
Do
Yeah.
that. Yeah. I
That's
can totally
perfect.
do this here. Just going back in time. And there you are. All right.
Are you sharing screen? I don't see that. Oh, perfect. Now I see that. Yeah, thank you. So next slide, please. My name is Chenchen. I'm a scientist in Mike Snyder's lab here at Stanford. So we are doing quite a bit of tissue mapping for the human intestine using different spatial assays. Next slide, please. So we are also part of the HubMap, and our job is to map the complexity of the small and the large intestine. So we do this mapping for 8 sites across the human intestine, and we have an array of different omics methods. This includes methods at the bulk level, but in the recent years, we have been transitioning to a single cell and also spatial technologies. And on the left, you see the team for our TMC. Next slide, please. And this is a tissue we are mapping, the human intestine. So it's a section of the intestine wall. You see the different layers from the left to right. So it's the human, it's the epithelial layer, and then the mucosa, and then the muscle layer. And the histology is fascinating, and it contains very diverse cell types. And our job is to find out what are the different cell types and their states, and how they are interacting with each other in this tissue context. Next slide, please. And the reason we are going from bulk to single cell is obviously some of you have seen that with the bulk, you clearly see it's a mixture of everything. And with single cell, we are doing it really separating all the different cell types. Now with spatial, we can start to see the architecture. And next. And we started with using spatial proteomics assays like Codex. So we are mapping the antibody targets, but now we are transitioning to spatial transcriptomics, which means we are mapping the RNA targets. The reason we chose this is because the RNA targets, we don't need to validate the antibodies. Essentially, we are going with oligos that's complementary to our gene of interest. And the number of targets is also higher than the spatial proteomics assay that we have. And we can look at gene regulations because RNA is more direct to the DNA. And we can also do multi-omic integration together with our Codex data. Next. And at a high level, there are 2 different methods that we have evaluated for spatial transcriptomics. So one camp is the sequencing based, and the other camp is the imaging based. The major difference is the spatial resolution and also the number of targets you can do with a sequencing based method. Essentially, you are transferring the RNA molecules onto the microarray, and the spatial resolution is limited, but you can capture all the RNA molecules of interest. With the imaging, you are only looking at the genes of interest. But the spatial resolution is much higher because you are essentially imaging the tissue section with a microscope. Next slide, please. So specifically, one method we tried is the stereo-seq. So this is the brief overview of the technology, but you are transferring the RNA molecules onto the microarray. So it's DNA nanoballs. The nanoballs are so tiny, it's only 220 nanometer in size. That's why you can do single cell resolution. And once you have this transfer, you can sequence out the RNA molecule together with a barcode for the spatial location. And here on the right, you can see one spatial map for our human duodenum and tissue sample. Next slide, please. And these are the cell types. Next slide, please. Yeah, these are the cell types we can map using the marker genes that's detected using the stereo-seq. So the idea is that the cell types, they have specific expression for some of the marker genes, and using these marker genes, we know what are the cell types. And the same principle can be applied for other spatial transdermics methods. Next slide, please. For example, we have tried the 10x xenium, and this is the imaging-based method, and the spatial resolution is much higher. As you can see, the whole tissue section on the top left, and then you can zoom in just to for the epithelial layer, and you can further zoom in to the much higher resolution for the microvilli structure, and with the different cells segmented out, using segmentation methods for the depistate. Next slide, please. Although we only have 377 targets for this particular panel, but because these targets are specific for the genes for the cell types of interest, so essentially for these marker genes that we know from our single nuclei data, so we can also map out the tissue structure together with the cell types in a very high detail. So these are all the cell types we can measure, and you can see their tissue structure. For example, the stem cell is really in the middle of the submucosa and the enterocytes, and this is important because stem cells are there to replenish our enterocytes when we lose them during the digestive process. Next slide, please. And we have applied now these methods for different sites of the human intestine. You can see the 10x xeniums methods. You clearly see across the intestine. So starting from the small intestine, the very beginning of the small intestine is the duodenum, and the ileum is in the middle of the small and large intestine, and the sigmoid is the very end of the large intestine. You clearly see their tissue structure, they vary, and the thickness of the muscle layer also, they vary. The green color shows the stroma, or the muscle cells, and this corresponds to their physiology function, and we are trying to study and to have a better understanding of the tissue structure and also their related function. Next slide, please. And yeah, this is a high-level summary. So we evaluated these spatial transdermics methods, and we have chosen the stereo-seq mainly for discovery because we can do on-target survey of all the RNA molecules. And the xenium, we just showed you that it's very good for data production. It's relatively robust and high throughput. And these two assays, they work with different tissue preservation methods, and it's good for whole tissue section. And we have evaluated sensitivity, specificity, and all these assays, but I don't want to put all the details here. Happy to take any questions. Next slide, please. And that's the team who, yeah, the whole group, yeah, it's a very diverse environment, and I'm happy to take any questions you might have. I don't know how much time I still have.
Thank you. This is great. And yes, you're part of an amazing team. You saw that there is now a new specification on how to do segmentation masks in HubMap. And so you can do single cell segmentation, FTU segmentation, anatomical structure segmentation. Are you planning to do all three levels?
We've done the single cell segmentation using either the vendor provided tools and also cell pulse. We are planning to use the FTU segmentation together with John Hickey with the Codex data and also for the xenium data. So we have not tried that yet. There will be something on the roadmap for the next year, so 2025. Yeah.
Super. Yeah, that's all supported now. All right. In the interest of time, to leave some time at the end, let's see who's next. I guess I need to go back to my website here. Kevin, are you ready for this?
Yeah, I'm going to go ahead and share from my side.
Perfect. This way I can see the chat. So I can't see it in the setup.
This out of the way. Okay, this is a fantastic opportunity. Thank you, Katy, for the opportunity. So we're going to be talking today about the oral and craniofacial structures, which I think are beautiful and wonderfully unique. We'll show a little bit of this, but I think there's a lot that can be promised in action on moving forward on this kind of data. So my name is Kevin Byrd, and I spent a lot of time as a clinician, and this is what I looked at for many, many years. I've since left the clinic and focused all of our efforts on single cell and spatial, trying to really understand the contribution of these cells to disease. And one of the things you can appreciate is that the functional units, if you will, in this case, these gross macrostructures that perform the functions of breathing and mastication, speech, identity, have all been organized quite distinctly in unique spaces. And one of the things that we know is that these these units of epithelium and soft and hard tissue, as they kind of exist in these spaces, are contributed both by stroma and epithelium in many of the sites. Those overlying epithelial layers are fed by a blood vessel or a kind of a neurovascular bundle system. And so these have been beautifully stained going back to the 1970s. This idea of spatial biology has always been inherent as part of oral biology and some of the structures that have been measured in these spaces. So we really wanted to understand, in collaboration with Katy and their group, some of the contributions to the neurovascular bundles, focusing mostly on vascular endothelial cells, and the contribution to the structures and health, and then we'll apply this to disease. One thing I'll at least highlight for this group, again, trying to keep this at high level for interest, is that the oral cavity is highly dynamic. I think following the last talk was a great introduction. This actually is a number of manifestations and distinct niches of inflammatory bowel disease in the oral cavity. This dramatically affects, usually through edema, which is coming from the blood vessels, to many sites across the oral cavity and severity of the disease activity. And so there's a lot of things, I think, to understand, not just locally in this space, but also in the connection of this space to other spaces throughout the body. And so one of the things we talk about is this bidirectional relationship of oral and systemic health. And so the goal of our lab is to appreciate at a cellular, molecular, and hopefully now at a tissue or meta-architectural level, the contribution of these sites to their own homeostasis, and the contribution of these sites to contribute to other sites for disease. The oral microbiome overlays a lot of these sites, and there's a huge contribution and relationship at a holobionic level of these sites. And so, as you may appreciate, that if we used to think of the oral cavity as one site, and now we think of it as many sites, at least in our lab, that what follows and what probably is lagging is a lot of cell culture models, animal models, and clinical trials that would be successful, taking into account this biology. And so the group has set out the last couple of years to create a heterogeneous map of all of these distinct sites, including both glands and mucosa. This is both public data we've generated, data we've integrated, and then data we've also moved forward on taking that information, moving into an integrated multi-omic atlas, including both spatial data of RNA and protein flavors, and also integrated data that includes cell-cell communication. And so we've built some tools to do this. What you can see at the top is mucosa and glands. What I'll highlight here in red is that contribution at that layer that we've now digitized, going back to those 1970s black and white images from old papers. Those red images are the vascular endothelial cells that are supporting this tissue, interspersed deeply within the glands. And then we've built tools to start to ask the contribution of these cell types to smaller relationships at a cell co-occurrence level. We're moving forward towards these defined, we call this cytocommunity, but we've done this at scale, so we call this constellation, and looking at the interaction of these functional tissue units that have been interspersed within tissues. And so going back to that idea of that old image, when we digitize, let's say this is a whole tongue we have here from top to bottom, left to right here is the tip of the tongue on the right side, the back of the throat on the left side. What you can see here is the contribution of these major vascular bundles that really have to be studied as they intersperse through muscle and move up towards the blue here, which is the epithelium. And so one of the things that we know is kind of this first collaboration with Ash and their team, and Katy's team at IU, is that the glands themselves are quite dynamic compared to mucosa. Even gland heterogeneity, as we look at different salivary glands and different mucosal heterogeneity, is kind of arising, and we're starting to try to understand this at a three-dimensional and a two-dimensional level. And so here in red, you can see the endothelial cells that are connected. The last thing I'll say, just based on this early data, is that when we start to look at the cell types we've defined based on these assays, what we call the coefficient of variation, we find that many of these sites have different variabilities in median distances based on immune cell types, which we think is quite relevant for disease and proclivities of some of these niches to break down in pathologies. And so if we use this data and we dive deeper and build spatial transcriptomic panels, we can eventually get to a place where we've locked in the vascular common coordinate framework and ask questions about cell-cell communication within local distances, and then map that back to some collaborations with Katy and their team, back to the exact site where we took the biopsy. One of the general availabilities as we started this talk in the beginning was that we can individually pinpoint the exact biopsy and then map that back to a 3D reconstruction of where these communicative events are, the cells, the molecules, and the tissues that contribute. So I look forward to thinking about this, not just in health, but a number of projects that are ongoing in the lab looking at both space in health and in disease. And so looking forward to sharing that with you all very soon. Thank you for your attention. Looking forward to the discussion at the end.
Kevin, thanks so much. I put the link to your new paper into the chat. Others might like to check that out. Glad it is out. Yes, just like Chenchen showed us the beginning of the small intestine, the middle part between the two small and large and the end of the large one. I think at some point we might have the entire digestive tract, right? You are in the beginning, Chenchen at the end. I think this will be super interesting to have. All right, next in line is Archibald. Would you like to share your own slides or should I run them? What do you prefer?
Yeah, I can share.
I think there's less of a delay if you do it.
Can you see it?
Yes.
So hi everybody, my name is Archie Enninful. I'm a PhD student in Dr. Wong's lab at Yale, and we're part of the HubMap TTD, where we're focused on the spatial multi-omics profiling of lymphoid tissues. And the specific tissue we are focused on is the lymph node. So the lymph nodes are the primary lymphoid, they're secondary lymphoid organs, and these are the sites for lymphocyte activation. Typically, they're found at the site of the convergence of major blood vessels. So for example, in the human body, you can find these in the neck or the axilla or the thorax or other parts of the body as well. And it has a very defined anatomical structure, but I won't go into too much detail. But there are very defined regions where you find B cells and T cells and other immune cell types. So as part of our work, we have 33 whole lymph node samples. 16 of these are FFPE and 17 of these are fresh frozen samples. And these samples, I mean, typically a patient would not give their lymph node in routine clinical work. So these are samples that are taken during, these are biopsies that were tested and found to be benign. So these are non-reactive lymph nodes. And they cover approximately equal representation for both genders. And most of our patients are primarily white patients. And in the context of the workflow, so we obtain the whole lymph node samples and then we get serial sections and we do routine histological stain, H&E, as well as codex, spatial transcriptomics, and spatial metabolomics using stimulated Raman spectroscopy. But for the purposes of this presentation, I'll focus mainly on codex. So this is just a snapshot of the histological stain from the lymph node samples we have. And our codex panel covers all the major immune cell types that you expect to find within the lymph node. And particularly, we also include CD31 as a marker for the endothelial cells, because as part of the vascular common coordinate framework, we would not want to know the distance of each cell type from the nearest endothelial cells. And we also included these 4 markers. These are related to cellular senescence, because we have another project also focused on the lymph node on cellular senescence, so we can also interrogate the process of senescence in lymph nodes and which cells become senescent. So here is just a representative codex image showing the different cell types. Here, we're just visualizing T cells and B cells, as well as the vasculature in red marked by CD31. So in terms of our data integration workflow, we perform cell segmentation using deep cell. It's a Mesmer-based method. So once we finish the cell segmentation, we proceed to annotate the codex data set. And to do this, we curated a single cell RNA-seq reference data set from multiple different sources, including the Tabula Sapiens and the SLO Atlas. So these have cell type labels, and then we integrate these 2 different modalities, which is single cell and codex, using this recently published pipeline called MaxFuse. So max views allows you to integrate modalities that are weakly linked. So from the single cell, we can transfer the cell type labels and annotate the whole codex data set. And once we do that, we can identify all the different immune subtypes, and their spatial locations match with what you expect to see in a normal lymph node architecture. So this is just one representative image of an annotated codex, human lymph node, and we can appreciate some of the structures. We see these really rounded structures. These are the B cell follicles that you expect to see in a normal healthy lymph node. So in the context of the vascular common coordinate framework visualizations, we are collaborating with Katy, Börner and her group, so we can measure the distance of each cell to the nearest vasculature. And for example, in the context of lymphoma, which is a separate project we're doing in our lab, this specific lymphoma called AITL, we find that the tumor cells tend to be very close to the vasculature, and that is very unique to AITL. So it's very interesting that we can, by measuring the distances of these cells to the immune, to the vasculature, we can gain more information, potentially even understand why that is the case. So I'd like to thank the whole Yale TMC, and I'll be happy to take any questions. ELTTD have up.
Thank you so much, Archie. You just generate the most amazing data, all of you, actually. It's just wonderful to see that data come into existence, because this is gold on the atlasing side. I wonder, so in Chenchen's slide, on slide 10 in this set, I saw that he was able to use different markers to identify microvasculature versus venules, and we would be happy to extend the cell distance explorer to actually indicate those differently, right? And then do different distances to those. I wonder if this is easy or hard on your end to basically have more markers for endothelial cells so that you can make that distinction.
We just need to include the markers in our panel. I think we also have CD34, but maybe Chenchen could chime in on the other markers they use for the vasculature. We could either conjugate them or purchase them from Akoya and include them in our panel.
Yeah, so we used RNA markers. I don't remember exactly the gene name for the vasculature. I think there was some PCAM or some, yeah, PCAM1. But I all need to double check the codex panel we have. So what I showed is not for the codex. You have the codex, right? No.
Yeah.
I can check now. Let me double check now and get back to you. Yeah.
Okay, thank you so much.
Yeah, any other questions before it goes over to Alex? All right, then Alex, it's up to you. So Alex is a postdoctoral fellow in the Sorga lab, and Sorga's team was able to share 3D data with us, super exciting. And so thank you for presenting here.
Thank you for the introduction, Katy. Yes, I'm a postdoc in Peter Sorga's lab at Harvard Medical School, and my work really focuses on developing a new 3D cyclical multiplexing method that integrates millimeter-sized human tissues and tumors, and specifically focusing on colorectal cancer. And so this method is, of course, based on the CySIF, an open source method. And yeah, this is just an image of the nerves in the colon stained with beta-3 tubulin. And you can see, so why do we need to understand like 3D features in cancer? And of course, as you saw, with not just vasculature and nerves, they actually go in and out of the plane. And so you can't really use this as a single plane representative of the whole tissue. Not only can you like study cell-to-cell interactions, but now when you go into deeper 3D, you can start to study the macroscale features and their interactions with the single cell. And you get this once you increase in resolution. As you go high in resolution, you also get the distributions. So as I mentioned before, those interactions, but also across the entire tissue. And then you can also capture very sparse features, because very coarse sampling would mean that you could actually miss those rather sparse events. And so in our lab, there's really 3 modalities of high-plex 3D imaging that's currently ongoing. The first is 3D zero section reconstruction, and that is using existing techniques, but taking adjacent slides. Although you could skip some of those slides, and it's very good for integrating different modalities like H&E or geomics. And so this was demonstrated in paper by Jerry Lin in 2023. And the second is with confocal microscopy, which allows you to see if with even higher resolution on a for now on a single section here, you can get the specific cell-to-cell interactions and also capture their morphology. And the third, which is what I'm working on, the light sheet microscopy, I want to marry the 2 benefits of zero sectioning across the large distances, as well as the high resolution, not as high as confocal, but like moving closer. And so one of the examples I wanted to show in Jerry's paper is that in 2D, you could see tumor budding cells, which are like most prognostic factor that you see about a cluster of 4 cells. But with zero sectioning, you're getting an approximation of the entire 3D tumor mass, and you're also not only seeing the change, like that they're actually interconnected structure, they're also having a epithelial mesenchymal transition-like phenotype. So there's a gradient happening in across this tumor. But actually, this was so conventional slides are actually only 5 microns thick, and it's like a slice of orange. You can't base the entire orange in this whole shape on just one slice, and so you can't really base that conclusion on single cell, because then what that means is that you're not accurately phenotyping those cells. And in this example, partial cells lead to inaccurate phenotyping. They're quite polarized. These punctate limited spots, such as LAG3 or granzyme B, they're actually polarized, could be on the top or the bottom of those cells. And so this could lead to a lot of inaccuracies. And then we're also integrating thick tissue 3D with the HugMap coordinate framework and also the cell distance explorer. And here in this was imaged on the confocal. Even a small branch of the captured vasculature is quite busy. As you can see, all the different cell types are interacting. But here you can also see that in 3D, because this kind of data size would get bigger as you move across to much larger tissue. And so being able to condense that and query this with the CDE will be very, very helpful. And here's a link to explore this data set imaged by Clarence. And so I talk a lot about light sheet and what are the benefits of light sheet. Not only is it much faster, it also covers larger distances, and you're not illuminating the entire tissue, which could cause photobleaching. The excitation is actually decoupled from the detection lens, unlike wide field and confocal. But this doesn't mean that we can immediately image much deeper. We also need to clear the tissue. We need to remove either some lipids or even just refractive index match, and basically turn it into a jelly-like substance and remove all the pigmentation to reduce the amount of light scattering and also light absorption. And so what you end up is with a transparent tissue. But the tricky thing is to keep this very fragile tissue intact, that you can still interrogate all of the immune cells. And so we have demonstrated this on a piece of healthy colon tissue and registered several channels together. So this is like a 7 by 5 millimeter tissue. Here I've registered a small ROI just for clarity. You can see all the nerves at the submucosa, and then also going up into the individual villi. Also see the insides of keratin and where they're distributed along along the villi here. But also we want to enumerate all of the cytotoxic T cells and how they're interacting with all of these nerves. And so this is just a small 3D model of the calculations. You can see a lot of these cells are actually quite close to these nerves, and we actually see them physically interacting. And so, of course, the next stage, we want to also do this with vasculature. And so here we've stained a small piece of tissue with ERG1, ERG1, endothelial transcription factor 1. And here you can see they're actually chock full of potentially immune cells, but this is another reason why we need to do immunofluorescence, so we can start to phenotype all of these distributed cells. And so with that, I'd like to thank everybody involved in the project, and also like to thank my PI, Peter Sorger, and Clarence Yap, and everybody in the team, Daniel, Yvonne, and our collaborators. Thank you.
Thank you, Alex. Beautiful data. And I heard that at some point it might even be visible in the organ gallery, right? In 3D, potentially. I think
Yes,
some of you are wrapped into that junior investigator project to bring
right.
some of this amazing data into a 3D kind of zoomable interface that shows.
Yeah, I really enjoyed the workshop that was hosted by Andy, and yeah, being able to interact with those cells in VR and pull them up and then reconstruct them is something we're in the works.
And it
Thank you.
is truly 10 to the power of 10 zooms,
Yeah.
right? It's amazing. And so there's a panel later today on how do you deal with that multi-scale zoom? And then
Yeah.
there's a panel tomorrow morning on why in the world do we need 3D and do we need it really? And when do we need it? So yeah, feel free to come back if you have time.
Looking forward to
listen
that.
the recordings, yeah.
Thank you.
All right. Any questions for Alex? I think Ravi is next.
Hello, everyone. Thank you very much for the invitation to come and present some of the work here at this forum. Let me just get this into slideshow.
Thank you for presenting from your computer. So much easier on my end.
Perfect. Yeah, so thanks again for the opportunity to present here. So I'm a research associate professor in Dr. Gloria Pryuber's lab at the University of Rochester Medical Center. So I've basically been involved in lung research pretty much since the first days of the Lung Map Consortium, and I really helped kind of get that up off the ground. And then eventually we ended up becoming part of the HubMap first iteration with UCSD, and then Gloria had gotten the lung portion of that project for HubMap 2. So really, it's a huge group of people that we've been working with for the past 10, 15 years to really get to the point where we're at now. And I'm an immunologist kind of at heart. So it was great seeing, the past couple of presentations, a lot of focus on the immune system. So I decided to take kind of this tact of really focusing in on a specific disease that we've been interested in, and it's specifically bronchopulmonary dysplasia. So we're in the Department of Pediatrics here at the University of Rochester, and have really been kind of developing more of these big data sets that then we rely on a bunch of the other folks in some of the data coordinating centers and the Hive to help us make sense of them. So basically what we have here is, in the case of the lung, there's obviously a lot of space that's in there, which basically creates a lot of problems in terms of trying to image, or there's a lot of proteolysis that occurs in the lung. So really trying to get high quality tissue has kind of been at the heart of what our research program has been. So of course, with the lung, you have, going at the multiple scales, going all the way from, looking at the trachea down to the bronchi and into the lung, and then all the way down into the alveolar region, which is really where the gas exchange occurs. So, you have the big vessels and whatnot that's going to really be pushing out blood at high volumes. But then you have to get down into really kind of one cell at a time, where in the alveolar regions, you have the oxygen coming in, the carbon dioxide going out, that basically will allow you to breathe, right? So if you don't have that, if you don't have a functional unit there, then that becomes problematic because you're not getting the oxygen you need for your basically to live. So within the lung too, the organogenesis is very important, especially as I'd mentioned, we're really focused on, in the beginning, we were focused on pediatric lung development and most specifically postnatal. But just to kind of give you an idea of where the window of viability is for the great clinicians and the neonatal intensive care unit, they really will resuscitate at anywhere above 24 weeks of gestational age. So really, that's basically the limit to where you're able to have a functional lung that will actually exchange gas and be really kind of that window of viability. So as I'd mentioned, you really have these blood vessels that are bringing in high levels of blood, and then it goes all the way down into the capillary, eventually into the alveolar region that will allow for that gas exchange. So kids that are born preterm basically will be put onto ventilators and will end up having hyperoxia mediated lung damage and barotrauma, which will lead to problems with increase in fibrosis, increase in inflammation, increase in the alveolar size, and basically lung damage. So what happens then is this disease persists into adulthood and basically ends up like a COPD type of situation where there's just problems all the way up into adulthood. There's also fibrosis that occurs, and there's also, in some cases, pulmonary hypertension because there's a backup of blood in the lung, and that then leads to an enlargement of the pulmonary arteries. So in a normal healthy bronchiole, you have nice airflow that goes in, you get nice gas exchange, and there are some of the structures like the capillaries that I had mentioned. So in diseases such as asthma, you can have basically obstruction of your breath in, breath out. And again, it's kind of a similar thing where you end up having poor gas exchange and potentially increased infections because you're not really able to get rid of the gunk that's in there. And then in bronchopulmonary dysplasia, you also have problems getting air in and out. And there's basically airway disease that also goes down into the bronchoalveolar duct regions. And on top of all that problem with being able to exchange gas, it just sets up for problems later in life. So as I mentioned, I'm also an immunologist really at heart. So you also have a bunch of these other cell types like the activated macrophages, neutrophils, T cells, B cells. They're all coming in, and if they're kind of in the diffuse category within that alveolar region, it will lead to additional damage. And that damage could be direct inflammation. It can also lead to damage of the fibroblasts that are within that alveolar region, leading to that fibrosis and that scarring. And also there's a lot of cell damage that occurs from oxidative stress from having all of those activated immune cells in there, which then leads to an enlargement of the extracellular matrix and really kind of a stiff lung. So this is an example of some work that we were completing with Katy's group and Yash at Indiana University, where we were focusing on studying bronchopulmonary dysplasia and healthy lung. So we have a relatively low end for this particular project for the mapping back of cells to endothelial cells, but we also have been expanding the number of slides that we have analyzed and are in the process of analyzing. So this is just to show in some of the samples that we've generated and analyzed using Codex that there's areas of inflammation. We see that enlarged perivascular muscle, we see thickening of matrix, and we see enlarged alveoli. So really kind of recapitulating what is in the literature. So using the phenocycler, and Dr. Jeff Perkerson, who is another research associate professor in our group, had generated an OMAP for generating, now we're up to over 45 different antibodies that will allow for us to get into subtypes of a variety of different cells, and also to map out the extracellular matrix and the thickening of that. So within this, this is that same H&E that I'd shown just a second ago, that now this is looking at one of the multiplex immunofluorescence, where you can see in the healthy lung, you have a pulmonary artery and a bronchus that are present, that are, basically have kind of that nice structure, not a lot of cells within there, and you have nice alveolar region. In comparison to the BPD lung, where now you're seeing in this particular case, there's that thickening around the pulmonary artery and the thickening of the extracellular matrix around the bronchus. So really, by being able to map how far away these cells are from each other, this is where Katy and Yash had graciously collaborated with Gloria's group and looking at just really where the distribution of the cells are in the healthy lung and the BPD lung. And in this particular case, you can see that there's quite a few of immune cells that are present within the BPD lung that's near vasculature. So this is basically now a way to be able to get into where these immune cells are in a disease state. And we use bronchopulmonary dysplasia as kind of the illustrative fact for the case studies that we're basically doing. And the idea then, we will be able to go into additional diseases and be able to map out where these immune cells are in relation to the vascular, not only the arteries, but also inside the alveolar regions where the capillaries are. And really kind of appreciate what the previous speakers have mentioned about punctate staining for some of these structures and basically bringing this now into 3D to be able to understand really where these capillaries are and where these large arteries are, to be able to understand what's going on with disease, not only for bronchopulmonary dysplasia, but for other diseases as well, such as asthma. And this is just kind of put in there to show that we are able to find many different structures. In this particular case, it was a 5-year-old, and this was a 30 antibody panel, but we've already gone up to, as I mentioned, over 45 antibodies. So it really does take a village to generate the data, and collaborations that have spanned now 10 to 15 years, and really taking it into the next level of being able to take serial sections of 2D codecs into 3D and then pass them off to folks like Katy's group to really kind of get an idea of what's going on in normal development and also in disease. So I will stop there and send it back to Katy.
Yeah, Ravi, absolute pleasure to work with you and Gloria's team. We learned a lot, and I actually have a friend, and they have a too early born child, and I see exactly the issues that you brought up in what they go through in their life, right? It goes on. It's not just in the early childhood. It goes on to being an adolescent and then from there. And so it's, I think, good to understand what is happening, and maybe at some point we can re-engineer it. So the presentation we just had was going in that direction, but I think right now we are just trying to do the inventory and trying to understand what is happening as we evolve in age. So I'm really glad that all of you are still here. And as you know, we are writing this joint paper so that we can start comparing our data across tissues, across the different technologies, across the different labs that are now all involved in, and trying to also then harmonize the data and link it to the cell ontology so that we ultimately can do that comparison. I wonder if there are challenges and opportunities you saw so far, and maybe also opportunities that you would like to do together after that one paper is done. Many of you attended the HubMap demo day where David Van Walen showed how he can now segment and annotate tissue, spatial tissue across different organs. And Rahul Zahid from the New York Genomic Center, he presented how pan-azimuth now can be applied across tissues and maybe even to tissues that has not been seen before. So I think all of this becomes possible now, and I wonder how far this advances your careers, right? Many of you are a junior investigator, maybe some of us are not, but many of you are. And it's really a unique opportunity for me to understand better what you would love to see in the next 5 to 10 years.
So I think I'll just very briefly jump in and say, I think really getting into true 3-dimensional would be really useful. And I think that, you know, we've done a little bit of multiphoton microscopy, but you can only get 4 colors in really one run that takes a long time. So that's where kind of this light sheet microscopy is really, you know, fascinating to me, and I know to Gloria's group in general.
And I wonder how much that costs, right? So we recently did that one exercise where tissue data, human healthy tissue data for the organs that have the functional tissue units that we are focusing in on, 22 across 10 organs, all were shipped to UC Davis, and then we're done with Phoebe Lightsheet, with Phoebe technology there, not Lightsheet. And that was super interesting because for the very first time, all data went through one technology and one team. And that's not typically the data I get to see. So I would love to see more efforts where multiple organs from one donor, just like the project that Gloria leads, get analyzed using either the same technology, Codex, let's say, or Xenium for that matter, because that seems to scale also very well now. But ideally by one lab, but it looks like you always have to also send postdoc to that lab, right? Because the tissues are so different.
Yeah, we have collaboration actually with Gloria and Sanjay's group. So we profiled lung and kidney, and together with our intestine here at Stanford with the Xenium technology. So I think for us, for me at least, that we would like to see more cross-organ comparison in the next 5 years. I think in the past 5 years, everyone is doing like focusing on their tissue of choice or their favorite tissue, right? And the question remains is that what are the different cell types that's common in all the different organs, and what are the organ-specific cell types, and what are the FTUs or the neighborhoods, how they are common, like for example, in the lung and then the intestine. We know that the lung and intestine, they are stem cells, they are immune neighborhoods, but how do they differ? I think these are the open questions that we would like to see for the HiMap, and also in general, like in the tissue mapping field in the next 3 years, that will be our top priority, to compare actually cross-organ with different technologies. One is the Xenium, and we also collected Codex data together with other groups. Yeah,
I think that's great. I think that the Xenium data looked beautiful. Gloria had shown a little bit of that, so it's really, really exciting times.
Yeah, we are starting to get into the insights of what the data means. I think it's still early days, but that will be the focus for the next year, I think. And together with Katy's group, we can also explore the cell distance from particular to the vasculature, and how they differ across organs. I think that will be very interesting for the HiMap. Yeah.
And as you go 3D, the distances actually get shorter, right? Because now cells that couldn't connect in the 2D, they now can connect to something that's above them or below them. So I think that's making for much better and higher accurate data.
I think I'll add to that. I think we're really excited to think about how it is that some of these spatial technologies and some of the common language of how the human body is constructed can be translated to, I think, clinical studies, especially those that involve biopsies taken both pre and post therapeutic intervention. I think the lung field has been tremendous in the way that they've led with precision cut lung slices. Some of those 6 slices, we've been doing this with some oral mucosa and some upper airway mucosa, and then culturing them for 7 to 14 days, challenging them with fibrotic inhibitors or activators. But we've gotten pretty good at putting them back into a block, cutting it, and putting it back onto a slide for a combined phenocycler and Xenium. And that's allowing us to see kind of perturbation networks that I think really, as the field is moving toward perturbation-seq and CRISPR assays and in vitro and in single cell. I think the spatial component of that will follow. And I think the work that we just saw here is truly a kind of a foundational element for moving forward on translating that both in young and old individuals.
And I know we're coming up here to the hour. As you might have seen, the Tabular Sapiens 2.0 is out. Yesterday in the demo day, we discussed a spatial Tabular Sapiens, right? How could we get to that? So multiple organs from multiple individuals analyzed using comparable workflows. And then, of course, also starting to align naming of cell types so that we can compare across. I think it's super exciting. And I was glad to see that the Nature Methods also named spatial technologies as the method of the year. So that is very appropriate. All right. Thank you so very, very much for coming here. It was really great to have you here on a Saturday afternoon.